Hello, in this video I'm going to be talking about hormones that are all made up of amino acids or amino acid derivatives and how they act on their target tissues. Specifically, I'm talking about hormones that are hydrophilic, right? That is, they are polar. They are made up of amino acid derivatives, made up of shorter chains of amino acids or very long chains of amino acids, sometimes with multiple subunits. There are many different ways that these hormones act on their target tissues, many different types of receptors. All receptors are, of course, very specific to the particular hormone in question. However, I'm going to show you today three different types of mechanisms of action. The first two we see quite often. The third one is a little bit less common. There are also a few mechanisms that I'm not going to go over today. They're a little bit less common, and I'm not going to be talking about them over and over again throughout the term. Again, focusing on cyclic AMP as a second messenger, IP3 and DAG, as well as tyrosine kinase receptors. The first mechanism I want to discuss today is cyclic AMP. Now, cyclic AMP is something called a second messenger. That is, the first messenger is one of the hormones, right, binding to a receptor and ultimately leading to the production of this second messenger. So it is not the hormone itself that is producing the pre-programmed effect throughout the rest of the cell. It is actually the second messenger, in this case, the cyclic AMP. And the reason why we use second messengers is to essentially amplify the response. One individual protein hormone molecule can produce very widespread, very large effects because of the increased production of cyclic AMP. Now let's walk through this process. First, the receptor for the particular protein embedded within the membrane of the target cell is called a G protein coupled receptor. Generally, these are 7 transmembranes. What we can see here is that there are pieces of the protein, right, the same receptor protein, that pass through the phospholipid bilayer that we can see here. And it does so 7 times. Now, there are portions of the protein that are extracellular. There are portions of the protein that are intracellular. Right? So again, this is a 7 transmembrane G protein coupled receptor, or GPCR. Okay? It is called G protein coupled because it is associated with a G protein, also called GTP. There are multiple subunits included within the G protein. And when the hormone binds to its specific G protein coupled receptor, one of these subunits, in this case the alpha subunit, is going to dissociate from the G protein, and it is going to bind to activating an enzyme called adenylate cyclase, also called adenylyl cyclase, depending on what source you're looking at. Now, this enzyme is responsible for taking an ATP molecule. And as we can see here, here's the ring of carbons, the sugar molecule, the nitrogenous base, in this case adenosine, and then here 3 phosphates. Right, so I will abbreviate phosphate as P, but this is actually a phosphorus surrounded by oxygens. And so ATP, this energy storage molecule, has 3 phosphates. Adenylate cyclase, once it is activated ultimately by the binding of the hormone, is going to remove the outer 2 phosphate groups, and this final phosphate is going to actually bind to the sugar molecule not just once, but twice. And so as we can see here, a phosphorus surrounded by 4 oxygens and bound to the sugar molecule of what was previously the ATP. Therefore, this molecule is called cyclic AMP. And so as long as the alpha subunit of the G protein is bound to the adenylate cyclase, this enzyme is going to continually convert ATP into cyclic AMP. Okay? So one molecule here, one hormone, is going to produce lots of cyclic AMP, lots of these second messengers. And so what exactly happens once we have these messengers? Okay. once again, the adenylate cyclase isn't only producing one CAMP, it is producing a lot of these molecules. And so here I have shown quite a few cyclic AMP. Cyclic AMP is going to bind to an inactive enzyme called protein kinase A, also abbreviated pKa. Okay. In this enzyme, there are 4 protein subunits. The first 2 subunits are regulatory. That is, they regulate when the catalytic subunits can actually catalyze reactions. In its inactive state, the regulatory subunits are bound to the 2 catalytic subunits, and therefore they're inactivated. Okay? But cyclic AMP binds to the regulatory subunits, and we can see here it requires 4 cyclic AMP. Okay, so not just a little bit of a signal, but fairly strong signal here. And the binding of cyclic AMP is going to liberate the 2 catalytic subunits from protein kinase A. So now they can go forth and they can catalyze a series of reactions. Specifically, kinases are responsible for phosphorylating proteins. There are different types of kinases that essentially add a phosphate group to different amino acids within proteins. And this can activate them, it can deactivate them, etc. Okay, so now that cyclic AMP has essentially liberated these 2 catalytic subunits, finally we start to get the downstream effects of the receptor, right? So whatever we would think of as the action of the hormone can be now produced by these kinases. Okay, so there are a lot of different things, and of course it depends very much on what particular cell we're looking at. Okay, this is just a general perspective here. So I'm giving you 2 different examples that are very generalized. This varies from cell to cell. The first action that the activated protein kinase A can have is that it can phosphorylate enzymes. Okay, so here we can see once again this ATP, 1, 2, 3 phosphates, the ATP, and an inactive enzyme. And again, this depends on what the cell actually is, what the effect of this hormone is. Okay? And one of these phosphates is removed and added to the enzyme. Okay? Ultimately, this is going to leave us with an ADP, right? The lower energy form of this molecule. The protein kinase A still activated, right? It's not bound up here to the regulatory subunits, and an activated enzyme. Oftentimes, this enzyme will go forth and it will activate another enzyme and another enzyme and another enzyme, producing something called a phosphorylation cascade. Okay? Another way that the activated protein kinase A works is it can enter into the nucleus and it can act as a transcription factor. Right? So essentially it can phosphorylate what is called Krebs on a promoter region of a particular gene. This promotes transcription, right? Therefore an mRNA copy of a particular gene, which will then go forth and be translated, processed, and then ultimately used. And so, to summarize, the hormone activated a G protein, which activated an adenylate cyclase, which produced a lot of the second messenger called cyclic AMP, which activated protein kinase A, which can phosphorylate enzymes, and it can act as a transcription factor. The second mechanism I want to discuss is called the IP3 DAG system. Okay. In this diagram, I have simplified a couple of the things a little bit because we have seen them before in the cyclic AMP system. This is that 7-transmembrane G protein coupled receptor. Okay, I've just simplified it by showing it as this big block here. Note that it is still associated with the G protein. Granted, the G proteins between the two systems are slightly different, but we are not going to discuss that today. Okay, so, once again, in this scenario, a hormone binds to this G protein coupled receptor, activating the G protein, right? One subunit dissociates, and it is going to activate this enzyme right here, which is called phospholipase C. This enzyme is responsible for breaking down a molecule that is embedded within the phospholipid bilayer. That membrane is this right here. Note these are two fatty acids. Okay, here is a phosphate, so essentially very similar to a normal phospholipid so far. And it is also linked to this ring of carbons with a couple more phosphates. This molecule is called phosphatidylinositol biphosphate, okay, or for short, Ip3. Okay, so once the hormone binds to the receptor, G protein activates phospholipase C. What this enzyme is going to do is essentially cut the two fatty acids off of this ring of carbons with its associated phosphates, producing two subsequent molecules. I will start with the first here. Again, ring of carbons with 3 phosphates. For short, this is called IP3. The full name of this molecule is called inositol triphosphate. Triphosphate, 1, 2, 3 phosphates. Now, IP3 has a series of different events. I'm just going to highlight a couple here. First of all, IP3 can bind to IP3-gated calcium channels that are embedded within the rough endoplasmic reticulum membrane. Okay. This, the RER, is responsible for doing a lot of things, including being a reservoir of calcium. So as soon as IP3 binds to these receptors, the associated ion channel opens up, calcium flows from its area of high storage concentration to its lower concentration within the cytosol, and calcium is going to act in a lot of different ways itself. We know that calcium can induce muscle contraction. Okay, so if this is, for example, a smooth muscle cell, the smooth muscle cell is going to contract, again, in response to hormone binding to its receptor. Calcium is also a strong promoter of exocytosis. So if this is some kind of secretory cell, the exocytosis of those contents can be triggered by, again, the hormone binding to the receptor. Calcium can activate a molecule called calmodulin to do a series of different activities, again, depending on the identity of the target cell. Calmodulin can activate kinases, right? We know from before that kinases phosphorylate things, and so the phosphorylation can lead to neurotransmitter secretion. It can act as a transcription factor, like we saw with protein kinase A. It can alter metabolism, specifically of things like glycogen. Okay? And finally, calcium can work together with this other component produced by phospholipase C. Up here, this is called activated protein kinase C. And the way that it is activated is the remaining 2 fatty acids from Ip3. Right? This molecule is called DAG, or diacylglycerol. Okay? DAG is going to interact with protein kinase C. Calcium, again, liberated from the RER by IP3. Calcium is also going to interact with protein kinase C, and together, both of these are going to activate this molecule. So PKC, protein kinase C. And once again, it's a kinase. It's going to go forth and phosphorylate things depending on the identity of the cell. One other note here, DAG, as 2 fatty acids, can also be used to create eicosanoids, so another type of cell signaling molecule. The final mechanism I want to talk about is a little bit less understood, but I do want to give you an example because we will see this for important hormones such as insulin. Okay? So what we can see here is not a G protein coupled receptor. Instead, this is called a tyrosine kinase receptor. Once again, we have multiple protein subunits joining together to produce this larger receptor. Okay? But this time, it is not associated with a G protein. Instead, when the appropriate ligand binds, in this case insulin as the hormone, what's going to happen is these components of the tyrosine kinase receptor are going to be phosphorylated. So here we can see ATP coming into the receptor in response to insulin binding. Okay? a phosphate is removed from ATP, leaving us with ADP as well as a phosphate group. And so this happens a couple times, and ultimately the phosphorylation of this tyrosine kinase is going to lead to the phosphorylation, or enable the phosphorylation, of various enzymes within the cell. Okay, so here we can see miscellaneous enzyme. Okay, it is phosphorylated, right, when this receptor here becomes phosphorylated. Okay, so this phosphate activates the tyrosine kinase receptor, and therefore this enzyme is going to be phosphorylated, right, on the tyrosines, therefore taking ATP, removing a phosphate, and leaving us with ADP. And so this diagram looks a lot simpler than all the others I've shown you so far, but in fact it is wildly complex. Essentially, the tyrosine kinase receptor phosphorylates this enzyme, which phosphorylates another enzyme, which phosphorylates another enzyme, leading to a true phosphorylation cascade. Ultimately, cellular effects such as the insertion of different channel proteins, such as glucose, GLUT2 channel protein, are inserted into the membrane, different genes are expressed, and so on and so forth. Once again, this has been a video about the three different examples of membrane receptors and their ultimate action in response to binding to different types of proteinaceous hormones, longer proteins, shorter chain peptides, and amino acid derivatives. And once again, cyclic AMP and IP3 DAG, both of these utilize are used as second messengers, and then we have the tyrosine kinase. Thank you so much for watching.